A lower cell count in the lower chamber does not automatically mean a compound has blocked motility. It may reflect reduced viability, altered adhesion, changes in proliferation, or a poorly controlled assay condition. That is why selecting between migration, chemotaxis, and invasion assays is a biological decision, not simply a choice of laboratory format. These assays measure related but distinct questions. A migration assay asks whether cells cross a porous membrane under defined culture conditions. A chemotaxis assay asks whether cells move preferentially toward a chemical signal, following a concentration gradient. An invasion assay adds an extracellular-matrix-like barrier, making barrier traversal part of the endpoint. The correct format begins with the claim the discovery program needs to test. If the question is whether a compound changes unprompted movement in a selected cell model, a transwell migration assay may be appropriate. If the intended mechanism involves recruitment toward a defined ligand, chemotaxis is required. If the hypothesis depends on movement through a matrix-like barrier, an invasion assay may provide the necessary additional evidence. Migration is useful for early phenotypic comparisons, dose ranging, and testing whether genetic or pharmacological perturbation changes motility. Yet the result remains specific to movement across that membrane under those experimental conditions. Cell density, pore size, serum exposure, incubation time, adhesion, proliferation, and viability can all influence the readout. The study should also state whether the endpoint relies on stained cells, fluorescence, ADP-associated signal, or image-based counts, and how background and starting cell differences were handled. Chemotaxis introduces a further requirement: a credible, biologically relevant gradient that remains informative during the incubation period. A lower chamber ligand alone does not establish directional movement. If the gradient dissipates too quickly, the experiment may increasingly measure chemokinesis, or non-directional movement. The cell population, chemoattractant, receptor biology, concentration range, and time window therefore need to be defined together. This is particularly important for receptor antagonist programs, where the desired conclusion concerns a ligand-receptor pathway rather than general cellular health. Invasion assays add an ECM-like layer before membrane passage. That barrier can make the format more relevant when invasive behavior or matrix interaction is central to the hypothesis. It also adds variables: matrix composition, coating consistency, barrier thickness, polymerization conditions, storage, and lot behavior. A reduced invasion signal may reflect altered movement, altered matrix interaction, reduced viability, or a combination. A barrier-free migration condition can help distinguish a specific barrier effect from a broader movement defect. Across all three formats, cytotoxicity is the central interpretive risk. The concentration range should be chosen so that lower movement can be assessed alongside viable cell number under matched exposure conditions. Vehicle controls establish baseline behavior. Positive controls confirm that the system detects an expected direction of effect, while negative controls clarify background. For chemotaxis, conditions that remove or equalize the gradient can help determine whether directionality contributes to the result. A decision-ready study also requires replicate consistency, traceable images or signals, pre-declared exclusion criteria, normalization details, and access to individual replicate data or another auditable form of raw output. 1% without experimental context is not enough to judge variability or justify follow-up work. The outsourcing brief should define the biological hypothesis, cell model, compound conditions, assay format, membrane and matrix parameters, controls, replicate structure, statistical expectations, and downstream validation path. Migration, chemotaxis, and invasion each resolve a different uncertainty. Their value depends on matching the assay to the claim, then keeping the conclusion within the evidence that the experiment can actually support.